Hello everyone, I am Louis Carl from EAVAG, the Swiss Federal Institute of Aquatic Science and Technology. I am working as a postdoc in the Ecolmpact 2 project, which is a multidisciplinary project involving researchers from several departments at EAVAG. Today I will present the result of the first part of the project, which was focused on the impact of treated wastewater effluent on Periphyton and the observed increased tolerance to micropollutants. Wastewater treatment plants can constitute the main point source pollution in stream by discharging an effluent rich in nutrients, micropollutants, and microorganisms. Among these constituents, one major concern comes from the micropollutants, which can impact the river ecosystem. Therefore, wastewater treatment plants in Switzerland are currently upgraded in order to remove micropollutants and consequently reduce the effluent toxicity. However, the challenge is to establish causality between exposure and effects of micropollutants on the biota. To achieve this goal, we use Periphyton as a biological model. Periphyton is considered as a microbial skin of the stream, and it consists in a complex assemblage of microbial communities including algae and bacteria which are growing at the surface of the river bed. Periphyton plays an important role in ecosystem functioning, and microorganisms forming Periphyton are potentially targeted by micropollutants. Indeed, some species in Periphyton are particularly sensitive to micropollutants, while some others can be quite tolerant. Our approach is based on the pollution-induced community tolerance concept. Let's take two different microbial communities as example. The first one, considered as a control, is not exposed or exposed to low level of pollutants in the environment. We can bring this community to the lab and expose the organisms to an increasing dose of the pollutants of interest. Then we measure their activity. We can be, for example, photosynthesis or respiration, and observe a decrease in activity with increasing dose of pollutants. From this dose-response curve, we can derive several parameters, the most used being the effective concentration 50%, EC50, which corresponds to the dose of pollutants inducing a decrease of activity by 50%. If we do exactly the same, but now with a community which was chronically exposed to higher concentration of pollutants in the environment, we can also obtain an EC50 value, but in that case, the EC50 value will be higher than the first one. Indeed, the second community is less sensitive than the first one, and we need more pollutants to induce the same effect. The higher the EC50 is, the higher is the tolerance of the community to the tested pollutants. So this is the concept, but what's behind this shift in tolerance? The chronic exposure to pollutants, as well as other important factors, such as nutrients and microorganisms, lead to physiological and structural changes in the community. For instance, the more sensitive species are eliminated, while the more tolerant ones are increasing in the community. In the environment, the presence of PICT could be considered as a warning system, as it reflects the exposure of microbial communities to micropollutants, and it's also the sign of changes in microbial community composition which can disturb ecosystem functioning. The induced tolerance of Periphyton to micropollutants has already been observed in the field, downstream wastewater treatment plants by using complex mixtures of micropollutants extracted from passive samplers that have been deployed at the wastewater treatment plant effluents. Moreover, this induced tolerance was lost after wastewater treatment plants upgrade with activated carbon, which led to 80% removal of micropollutants, indicating that PICT could constitute a useful tool for linking pollutant exposure and community-level effects. However, in the field, it's harder to understand the underlying mechanisms of induced tolerance by micropollutants in Periphyton communities. The project Ecolmpact 2 aimed at understanding these mechanisms. We used Periphyton as a biological model and tested its tolerance to micropollutants from the wastewater effluent. This project has four main questions. Can we observe PICT in Periphyton growing in experimental flumes that are treated with wastewater treatment plant effluents? Is the observed PICT in Periphyton downstream wastewater treatment plants driven by micropollutant exposure or by the release of tolerant microorganisms from the wastewater treatment plant, or a combination of both? To which degree do different advanced wastewater treatment technologies affect the tolerance development to micropollutants in Periphyton? And what's the potential role of PICT for future monitoring to assess the biological effects of upgrading wastewater treatment plants? To answer these questions, in the project we combine experimental studies in artificial stream under controlled conditions with field surveys. Today I will present the result of the first experiment, which aimed at answering the first question and which was done in the artificial stream system called Maiandros. Maiandros consists in 20 artificial flow-through channels mimicking the river, where Periphyton grows on glass slides under different mixtures of river and wastewater effluent. The entire system is mobile. It can be installed in other wastewater treatment plants. The system is continuously alimented with river water directly pumped in the stream and treated wastewater effluent. An online monitoring system allows controlling the mixtures of river and wastewater, as well as monitoring a set of water parameters such as conductivity and temperature. The river and wastewater effluent are mixed according to our experimental conditions, and the channels are continuously alimented with the different mixtures. In this experiment, we used 16 channels alimented with 4 different mixtures, 0, 10, 30, and 80% wastewater. We had 4 replicates per treatment, one channel corresponding to one biological replicate. Periphyton was grown on glass slides in the channels for 4 weeks, and you can see here the glass slides colonized by Periphyton in the channel. A set of biological endpoints, including microbial diversity, were measured at the end of the experiment. Moreover, we took water samples to determine general water chemistry and the concentrations of 51 micropollutants. Wastewater samples were also taken to determine microbial diversity. So let's start with the results of micropollutants analysis. Here are presented the concentrations of the analyzed micropollutants in the water of the artificial streams for the 4 experimental conditions, 0, 10, 30, and 80% wastewater. First of all, we could clearly see that the total micropollutant concentration in the wastewater was about 20 times higher than in the stream water. Then we could also observe a gradual increase in micropollutant concentrations in the channels according to wastewater proportion. Finally, the results also highlight the predominance of artificial sweeteners and pharmaceuticals, which is typical for urban wastewater effluent. And as we can see here, the presence of micropollutants was also confirmed in Periphyton, which is known to be able to bioaccumulate micropollutants. Similar gradual increase with wastewater proportion was also observed in Periphyton. However, the relative proportion of some compounds was considerably different in Periphyton than in the water, for example, the artificial sweeteners. This could be explained by the specific properties of the chemicals of the Periphyton matrix, and also probably by several processes such as active uptake and elimination in the Periphyton, as well as biotransformation. Nonetheless, the results clearly show that the Periphyton were exposed to increasing concentrations of micropollutants. So what does this mean for the tolerance and diversity of Periphyton? We assessed the induced tolerance of Periphyton to micropollutants at the end of the 4-week colonization period based on the PICT approach. We harvested Periphyton from each channel, scrapped them from the glass slides, and suspended them in mineral water. Each biofilm suspension was then exposed to increasing concentrations of micropollutants extracted from passive samplers deployed during the colonization period in the wastewater effluent. We measured 3 biological activities, 2 targeting phototrophs and 1 targeting heterotrophs, secondary production. However, we did not observe any inhibition of the secondary production, but we obtained dose response curves and EC50 values for each river wastewater mixtures only for the 2 phototrophs endpoints. And here are the EC50s that we obtained, either by measuring photosynthetic efficiency or primary production. The results are expressed as relative dilution factor in comparison to the pure micropollutant extract from passive samplers. We could observe a significant increase in EC50 for 30 and 80% for the photosynthetic efficiency and 80% for the primary production, meaning that these Periphyton were more tolerant to micropollutant from the passive samplers than the control ones. These results confirm that wastewater induced the tolerance of Periphyton to micropollutants, and therefore that PICT is reproducible in control conditions. We answered yes, the first question of the project for the 2 measured endpoints. Now the question is how this increased tolerance relates to changes in microbial communities composition within Periphyton. We looked at the genetic diversity of prokaryotes and eukaryotes by sequencing 16S and 18S rRNA genes, both in Periphyton communities after the 4-week colonization period and in wastewater effluent along the experiment. Here are the results of the beta diversity for both prokaryotes and eukaryotes for the Periphyton exposed to the 4 mixtures of river and wastewater, and for the wastewater. As you can see, control Periphyton in blue and wastewater microbial communities in black are well distinct. This is not surprising since they come from 2 different environments and correspond to 2 different lifestyles. What was really interesting was the clear changes in Periphyton communities in the presence of wastewater. Moreover, the changes was gradual and followed wastewater proportion. This change is likely due to the presence of nutrients and micropollutants, but it can also result from the colonization of Periphyton by microorganisms from wastewater. If we have a deeper look in the community composition, here for the prokaryotes, we could identify an effect of wastewater, for example here at the phylum level, with an increase or decrease in abundance with increasing wastewater proportion. This absence of clear pattern regarding the impact of wastewater effluent on prokaryotes could partially explain why the secondary production was not inhibited by the micropollutants in our bioassays. However, we could see a strong increase in cyanobacteria abundance in Periphyton with increase in wastewater proportion, suggesting their potential implication in the development of tolerance. And here, for the eukaryotes, again, the abundance of each phylum was differentially affected, either positively or negatively by the increase of wastewater effluent. In that case, we could clearly see a strong decrease of the abundance of diatoms, reflecting their sensitivity to the increase in wastewater, which can be explained by several factors like increase in nutrients and or micropollutants concentrations or negative impact of microorganisms from the wastewater. In conclusion, for the first part of the project, we could see that based on two phototroph endpoints, PICT is reproducible in artificial stream system. Treated wastewater increases the tolerance of phototrophs toward micropollutants. Treated wastewater also induced changes in the structure of prokaryotic and eukaryotic communities following wastewater proportion. And the abundance of cyanobacteria was increasing in Periphyton with wastewater proportion, the opposite being observed for diatoms. Today I only presented the results of the first experiment, but we also performed a second experiment in order to determine the potential role of tolerant microorganisms released in the wastewater to the observed increased tolerance of Periphyton to micropollutants. To do this, we implemented the Maiandros stream system with a filtration unit in order to remove the microorganisms from the effluent. We are still working on the results, which look promising, and several field surveys will also help answering this question by comparing the tolerance of Periphyton upstream wastewater treatment plant and several downstream sites to see how peaked the microorganisms and the micropollutants are conserved along a longitudinal transect. I hope I can present physically these new findings during the next Ecotoxicomic conference. With this, I would like to thank all the people involved in this collaborative and multidisciplinary project. Thanks to the organizers for this virtual edition of the Ecotoxicomic conference, and thank you for watching my presentation. I'm looking forward to the upcoming discussion. Goodbye and see you soon on Zoom.